Thanks very much. Very glad to be here today, and I want to talk to you about basically the work that we do and that we have been doing for the last 15 years or so. I fell in love at some point in my career with the forward genetic approach, which means starting with unusual phenotypes, exceptions to the norm, and finding the molecular cause, at which point you know something about how a certain biological system operates. Lately, this has become accelerated tremendously by advances in engineering, specifically the ability to sequence much faster than ever before, and also advances in computation, because it's a major challenge to process sequence once you have acquired it, and to actually find mutations from among the billions of base pairs in the mammalian genome. I'm motivated in all this work by a medical problem, although it's certainly the methods I'll talk about are applicable to many different medical problems. It's a fact that in the world today, about 1/4 of all people who are born will ultimately die of infection, and that means close to 15 million people every year. There is something special about infection too, and as we see in the developing world, and we have to remember all of the world was the developing world a rather short time ago, the majority of deaths, a clear majority of deaths, are caused by infection during ages prior to reproduction. And this means that over time, in recent times, and certainly in much less recent times as well, infection has been a tremendous selective pressure on our species, and it's very much shaped our genome, and it brought our immune system into existence. It was this intense selective pressure that made a very complex system of cells and molecules that resist infection by viruses, bacteria, fungi, parasites, any foreign life form that we might come in contact with. There are probably thousands of genes needed for comprehensive immunity to all these pathogens, and at present we don't have a complete list of what those genes are. We do know that failure of immunity, de facto failure as defined by death from infection, is not rare. And there have been hundreds of inherited immunodeficiency diseases described, but certainly we don't know all the causes of immune deficiency, and we don't have a very vivid picture of how the immune system operates. The flip side of having an immune system is that it's dangerous. We all can be prey to inflammatory or autoimmune diseases. Once you have this powerful system that attacks microbes, it sometimes is turned against self. And very common diseases like systemic lupus erythematosus at the lower right, or rheumatoid arthritis at the upper left, type 1 diabetes, autoinflammatory diseases like gout, all are caused by immune cells in the end. We would like to think of the immune system as a very complex machine with its different parts distributed across different cells and tissues. And our first step in understanding how immunity works would be to have a comprehensive list of the essential parts of that machine. Genetics is a wonderful tool for finding a list of parts like that. One can dissect any complex biological system using random germline mutagenesis. The principle is that one keeps a certain phenomenon of interest under surveillance while one damages genes at random and finds phenotypes, which are exceptions to the normal behavior of the system. And it's then possible to track down the cause of those exceptions and to find the genes that are critical. If you reach a state where all genes have been heavily mutagenized, saturation, however you define it, then you can be confident that you have a comprehensive list of the essential genes for a certain phenomenon. So this approach is akin to reverse engineering. One applies it to a living system, and one learns what's important by making the system fail over and over and over again in different ways. The advantages of forward genetics as a tool are as follows. First of all, it's unbiased. You don't begin with a hypothesis, and often the phenotypes that you see constitute big surprises. One can use the approach both to create disease or to suppress disease, and both approaches are informative and they can lead directly to therapies. If you pursue mutagenesis in depth, as I just indicated, you can reveal essentially all parts of a biological system. And also extreme phenotypes that one encounters in the course of this work can give insight into how the whole system operates, how relatively more common aberrations of a milder degree come about. But the problems with forward genetics were that historically it was quite difficult and slow. Often years were required to find the cause of a phenotype. And the main impediments were the lack of a genome reference sequence. We didn't know the gene content of the mammalian genome, and also low sequencing power slowed things down a great deal. The computational tools for rapid mapping and identification of mutations were also lacking, and there was no way to monitor saturation. We were proceeding blindly when we mutagenized the genome. We had no inkling of just how much damage we had done in a given set of mice, for example, that we might look at, and this too was something that was bothersome. Now today things have changed very much, and that's what I'm mainly going to talk about. We're currently limited only by the speed at which phenotype can be produced and assayed. And in fact, we can speak of positionally cloning mutations in real time. As soon as you see a phenotype, you know the cause. And that is exactly what I'll tell you, how that works and how it's come about. Even relatively weak phenotypes can be ascribed a genetic cause as well, using the methods I'm going to describe. And saturation is monitored concurrently with mutagenesis, so you know where you stand, whether you've destroyed 5%, 10%, 20% of the genome, just how much you can expect to have identified of the machine. It's also possible to categorically state that certain genes are not involved in a certain phenomenon of interest, and that's valuable too. The tool that we use for creating phenotype in mammals is a point mutagen called ethyl nitrosourea. And all of the aberrant-looking mice that you see here were created with ENU. Some of them have problems with coat color or with the quality of the coat. Others have behavioral problems. And one can imagine that immune deficiencies are also created in such a collection of mice. The process begins with the injection of ENU into mice with a known background sequence, homozygous at all loci, and these are C57 black 6 animals. ENU is an alkylating agent, and it will form adducts with DNA bases within spermatogonia. The majority of spermatogonia in male mice to which it's administered are killed, but the surviving population will grow back to repopulate the testis, and thereafter the sperm made by these mice have a certain burden of mutations. We know, in fact, what that burden is. We know that in the average surviving spermatogonial cell, there are an average of 134 mutations that cause coding change. And that's a very precise number, and I'll come back to it a bit later and tell you how we know it. We know also that virtually all phenotype results from changes in coding sense and not from remote intergenic changes in the genome. So these 134 mutations are distributed all among the chromosomes, which are each long single molecules of DNA. And in the process of meiosis, only half of the mutations are transmitted and packaged within each sperm. 67 mutations then, on average, are transmitted to all of the progeny of this male mouse when it's bred. Typically, we mutagenize 2 G0 males, as we call them. The first one is bred to produce a daughter that's heterozygous for an average of 67 mutations. The second one is bred to that daughter, and we get then the G1 mouse, which carries an average of 91 mutations. That mouse is crossed to another black 6 female, and on average 45 mutations are transmitted to each of the offspring, and G2 daughters are back-crossed to the G1. In all, we make a total then of about 50 or so mice per pedigree, and every pedigree has one and only one G1 mouse, and that's how the pedigree is defined. On average, every one of the mice in this G3 generation, which is screened for phenotype, will have 68 mutations, and 11 of those on average would be in the homozygous state, but somewhat fewer than that is observed because of lethality effects. The G1 mouse is particularly important for all of our studies because it's this mouse from which we archive sperm, so we save all the mutations that we make in a frozen form and can retrieve them at any time. And we also do whole exome sequencing to determine exactly what mutations are present in this mouse. Whole exome sequencing is done by enriching the coding part of the genome and then sequencing it in depth. And this is done on a HiSeq 2500 Illumina sequencer, which can deliver about 1 trillion base pairs of sequence every 3 weeks. It's kind of humbling to realize after the last talk that this is only 1/300th the rate at which Procter & Gamble makes diapers, but it seemed like a lot to me. In any case, these mutations, of course, have to be processed, these sequences have to be processed. All of them are short reads, and they have to be mapped to the reference genome sequence, which requires quite a lot of computational power, though certainly a manageable amount. The actual throughput is 0.6 trillion base pairs per run of 11 days, and then there's time required to start the machine again. So that trillion per 3 weeks is really a true figure allowing for downtime between runs. The size of the exome, as we define it, is 52 million base pairs, and we run typically 80 exomes, 80 mice equivalents, every time we run the machine, and the average depth of the reads at every base is 140 reads. Of course, there's some variability, and some are not covered to that depth, but we know that 96% of all target bases are covered to a depth of 10 or greater, and that means that the discovery of point mutations has about that same efficiency. We're identifying about 96% of them. How do we know how many mutations there are? Well, of course, we've validated the mutations identified in high throughput sequencing. We know the level of accuracy. We know the number of true mutations found in every exome. And this is an indication of the number of mutations per exome shown on the x-axis, and the number of sequenced exomes on the y-axis, and the mean number is 67, which is how we come up with the number I used earlier. If we do this double pedigree construction that I described, we come up with a mean of 91. And now we're beyond 1,000, and those numbers haven't significantly changed. By this stage in our work, which has been done mostly in the last 12 months, we've accumulated a total of 90,000 mutations that we've archived, and these fall into 18,540 genes, which is 81% of all the protein encoding genes in the mouse. And some contribution has been made by the Goodnow lab at ANU, which is involved in this project with us. A total of 6,446 mutations are overtly null. That is to say, they're nonsense codons that terminate the protein chain, or they're splicing errors that would be assured to disrupt the production of a normal protein. And those fall into 4,672 genes, which is to say we've outright destroyed the function of about 20% of protein encoding genes during this last year. But we have to remember that many missense alleles are functionally null as well. And in fact, we have a rule of thumb that for every phenotype you observe that's caused by an overt null allele, there are 2 caused by a missense allele. And we believe overall that about 1/3 of all genes in the genome have been destroyed in our collection of mutations. By the way, we encourage other people to use these mutations, and any of you who are in this field who might want null alleles of any of the genes that we have, you can find them on our website very easily. Go to mutagenetics.utsouthwestern.edu and click on incidental mutations, and then enter the name of the gene you're interested in, and you'll quickly see what we have. We then screen the G3 mice while all of this is going on for phenovariants. And we have in all a total of 5 classes of phenotypic screens, more than 40 actual screens in all, and every mouse is screened with all of these screens. And I'm going to discuss today some adaptive immune screens. We're asking a simple question: what genes are essential for an antibody response in the mouse? And when I say antibody response, I'm talking about three different kinds of antibody response, three different kinds of immune challenge, two of which are dependent on T cells and one of which is T-independent. We have a certain picture to base our work on, or to fill in, let's say, or in some cases to dispute. We know that where antibody responses are concerned, if they're T-dependent, they depend on T cells. T cells are born in the thymus. The T cells have to be activated by antigen-presenting cells, mainly dendritic cells, and then they undergo mitogenesis and they expand to form a clone with specificity for the antigen. There's TB collaboration within lymphoid follicles, and follicular B cells then are induced to expand. These cells then have a choice of either becoming early antibody-secreting cells or of becoming memory B cells and undergoing high affinity maturation within the germinal center to give rise to high affinity antibody-secreting cells, or long-lived plasma cells. This is the way people generally think of the adaptive immune response, notwithstanding that many of the molecular components are not known. Our screen is fairly simple. We challenge the mice with three different injections of either T-dependent or T-independent substances. We give them either recombinant Semliki forest virus vector that encodes beta-galactosidase and then bleed them to look for antibody response. Later, we also give them NP-ficol. We also give them ova-alum, and then a second bleeding is done to look for responses to these antigens. We also do flow cytometry to see whether there are developmental problems with the production of lymphoid or myeloid cells. And to date, the screen has been carried through different numbers of mice for the different components, but for most of it, we've looked at more than 10,000 mice by this time. To summarize what we've found, here we have a list of all the phenotypes. In orange, you see phenotypes where the mice didn't make a normal antibody response to beta-galactosidase. In green, a failure of the T-independent response to NP-ficol, a long polymer with a hapten attached to it. In blue, a failure of the response to ovalbumin. And you see that there is overlap between the phenotypic categories as illustrated by the Venn diagram. Using a mixture of different techniques, and this has been something in progression, we've managed to track down quite a number of those mutations, and all the mutations found are illustrated by black dots. In all, we can say from this aspect of the screen, there are 174 phenotypes that we've found as of this week. 38 causative mutations were identified in 34 genes so far, and some of them are new and surprising, and some of them validate what's known. At the same time, we do the FACS screen, and this is shown in red on the left, and I've combined all of the 174 phenotypes that we identified in the antibody screen on the right, and here too we've had some success in finding things. If you take both of the screens together, and you see there's overlap between what's found, there are 224 phenotypes in all, 59 causative mutations identified. Those fall into 51 genes. These statistics should be viewed in the context of the fact that we've now achieved something like 15% of saturation of the genome. That is, we believe that 15% of all genes have been mutated to phenovariants. We know that we found only about 26% of the pool of available phenotypes, and on that basis we might estimate roughly that there are something like 1,300 genes with non-redundant function in the antibody response and/or development as defined by our screens. But many of these phenotypes were solved, those that have been solved, by rather old-fashioned methods. And I want to show you how we do it today, because things have changed and we're able to find the cause of phenotypes in real time. These days, when a phenotype is seen, immediately we know the cause, within the same minute or two. And here's how it works. We no longer screen mice and find a phenotype and say that's very interesting and go back and recross the mouse. Instead, we look at phenotypes, rather large phenotypes with 45 or 50 mice in each one. And here's an example of that. Here we're looking at the antibody response to beta-galactosidase triggered by injection of this recombinant Semliki forest virus vector. You see in the left column a bunch of control mice, and they all make an antibody response, and we take the mean of that response and designate it as 100%. On the right, we see a certain pedigree derived from the G1 mouse R0071, and you can see right away that there's something unusual about that pedigree. You see that indeed there are some mice that are pretty much in the normal range, but there are many that make an abnormal response, not a null response, only a few are down at 0, but quite a lot of them have an intermediate response of some kind. And the question is, what's causing this? When we see such a mutation, we typically give it a name. In this case, the name was Amber. And in the way we do things now, the G1 mouse had already been sequenced long before the screening was done, and we knew that that G1 mouse was contributing a total of 79 coding mutations to its descendants. And the question is, which of these mutations is responsible? There are 45 G3 mice with genotypic variation at those 79 sites. Unfortunately, these days, we also genotype all of those sites in all of those mice up front before any phenotypic assessment is done. This amounts to 3,555 sites in all. All of the G3 mice were genotyped using a new technology called the ion torrent sequencer. And this is how that machine works. It's very much smaller. It's a benchtop instrument. It has a throughput of about 1 gig of sequence every 6 hours, and then has to be recharged. And typically we would run the pedigree on this machine. We would genotype all of the mice at all of those sites using a multiplex PCR assay. And the data, when slightly processed, would come back to us like this. This is an Excel file, and it's color-coded. What you see is that these are all of the mutations, all 79 of them. These are each different mice in the columns, and at every site we know whether there's a homozygous variant in light blue, a heterozygote in green, or the reference sequence in homozygous form shown in yellow. A computer then instantly addresses these data, and really in the blink of an eye we're able to tell which mutation among those is responsible. A statistical assessment is made as to whether the assay score is correlated with genotype at every site, and recessive or semi-dominant or dominant models are all tested, and what we get back is a linkage plot that looks like this. So all this happens again in real time, and what you can see here is every mutation represented by a point, and the curve is discontinuous because we're looking at different chromosomes that have mutations. And we have 2 dotted lines here. This is the 5% significance line. This is the Bonferroni correction line. And what you can see right away is that only one of these mutations stands out. That's a mutation in aldehyde oxidase 3, which has a missense error. And this mutation we believe to be causative of the phenotype. The chances that it would have such a strong correlation by chance are less than 1 in a million. If we look at that particular mutation and we assess the antibody production performance of homozygous variants or heterozygotes or wild-type animals, we find the distribution of phenotypes to be just as shown, with the variants standing out. There are fewer of these variants than one would expect, and it's very likely that the mutation also compromises survival. It's also clear that this mutation is semi-dominant. It's not a purely recessive phenotype, and it might have been that by the old way of assessment, with only 1 or 2 mice detected, we would not even have detected the phenotype, and we would probably have not been able to map it so easily. At least it would have taken a very long time. So mutation finding has become a real-time process. When a phenotype is detected in any screen, whether it's a mouse that has a neurological problem or a coat color problem or an immune problem, then immediately we know its cause, or we can dismiss the phenovariance as non-genetic in origin. We can examine about 1,400 pedigrees per year, and that means that we're looking at something like 100,000 coding changes per year. And we're limited only by the rate at which we can generate mice and the rate at which we can screen. We also are able to estimate saturation from day to day, and we're even able to exonerate genes as being not involved in the process. In effect, what we've done has been to find several new parts of the machine. And the question is, where do these parts fit in? We can refer to our model, and I can tell you that some parts of the machine really sort of break the model in certain ways. But just to take a few examples, we know of a new transcription factor called ZBTB1 that's absolutely necessary for thymocytes to develop, or for all lymphoid cells to develop under certain conditions. We know that RUVBL2 is partially required for lymphoid development of both T and B cells. We know that ZEB1 is needed somehow for affinity maturation. It's another transcription factor. NF-κBID is a nuclear protein, a non-classical I-kappa-B-like protein, and it makes the decision, or at least is required for the decision, as to whether the expanded B-cell clone will go the route of becoming a short-lived antibody-secreting cell, or whether it will instead undergo affinity maturation and become a high-affinity plasma cell and contribute to the memory cell pool as well. Many of the mutations we find are simply confirmatory, and there's nothing wrong with that, and they simply tell us that our method is quite effective, and we can see the day when we can saturate the system, in fact, not so much in the future. We can project that half saturation of the genome might occur within the next 2 years and 80% saturation within the next 5 years, and it's our goal to archive a total of 1 million mutations for immediate retrieval. It's relatively simple to place these proteins on a high-altitude map. It's rather harder to establish precisely how they work, and that requires tools of biochemistry, cell biology, and structural biology, and a lot of strength has been built in that area as well. Some of the new genes encouragingly suggest ways that one might activate immunity, for example, for the purpose of making much more effective vaccines. Others suggest ways that one might dampen immunity for the treatment of autoimmune disease, for example, and these findings in many cases open the way to drug development. All the work I've told you about was done by this group, and in particular the screening for adaptive immune defects has been carried out by Ming Zhang, Scott Wang, and Jin Choi in my lab. We've also collaborated not only with Chris Goodnow, whom I mentioned, but also with Gunilla Carlson-Hedestum, Jerry McInerney, both of whom are at KI, and with Yang Si and Tao Wang at UT Southwestern to develop the instant statistical assessment programs that I showed. So thanks very much.
I have a question. Thank you. That was a really, really fascinating talk. I'm curious, are these sterile mice? Or do they not have any bacteria living in or on them?
No, they're not germ-free. They're maintained under specific pathogen housing conditions, specific pathogen-free housing conditions, but they're not germ-free.
Are you guys working then on how the bacterial and microbial community changes as a function of these mutations? I think that's another piece.
I think that is another screen, and it's one that could be done, again, by just use of brute force sequencing. And I think we're not quite there that we could go into depth and look at the whole microbiome of every mouse, but perhaps quite soon that might be possible.
Thank you. As I'm sure you know, we're seeing a similar, almost parallel explosion in our knowledge of human genes, where we have the single gene defect linked to phenotypes even as complicated as what you have here. So my question is, how much are you seeing a trend of, instead of discovery through your phenotyping leading you back to the gene that you're seeing, the discovery is a human counterpart, and then yourself or others coming to you and asking you, do you now have a gene model to satisfy the need to research further the human counterpart?
A lot of the known genes that I think I showed in red on that next to last slide were discovered originally in humans, and not so much in the recent times, but in more distant times. So it does happen. It's not the majority of what we find, though, still.
Bruce, that was a beautiful talk. Something struck me, though, especially in your last few points, that amazing discovery tools, amazing way to approach forward genetics, and I'm a big believer in those methodologies. But as you also know, sort of subject to the law of diminishing returns, where as you keep trying to mutagenize and you want, as you've stated, a complete list, a complete part list of our immune system. So are other technologies, CRISPR-Cas or other things that are emerging right now, helping you to orthogonally attack the set that seems to be less subject or you've not retrieved yet through the mutagenesis?
Yes, no question we hit large genes first, and there is a definite bias in what we hit when we use a point mutagen, and we've discussed, and we may go forward with it, using the CRISPR-Cas system to deliberately target very small genes that we know we haven't hit yet, and simply to do a certain number of them as mixtures injected into eggs, and to work our way through the list from small toward large that way. So I think that would be very useful to do, and to accumulate them and possibly regenerate them to do deliberate targeted screening that way.
Dr. Beutler, thank you for that terrific talk. Very fascinating, and I'm honored to be in the presence of a Nobel laureate. The reason I speak to you today is to ask you, with this fascinating accumulation of technology, which has rapidly expanded the ability for yourself and your other fellow researchers to do this type of work, what sort of tools do you expect or would you like to have in the near future to continue to accelerate and expand the type of scientific inquiry that you all are involved with?
I think it isn't so much a matter of tools. I will say it's sort of at the edge of what we can afford to do, because sequencing is not completely cheap yet. It's 1 million times cheaper than it used to be per base, but it still is expensive to pursue this whole approach of instant mapping by sequencing up front. So I'd like to see sequencing become even, let's say, 100 times cheaper. That would help. I think also it isn't so much a matter of developing new methods or technologies, but a great deal is centered on the screen and the cleverness of the screen. And we're only doing a certain number of screens, and we will get to a certain point, not absolute saturation, and we will move on to other screens. But I think that's where the real opportunity for creativity comes in this business.
Thank you, Doctor.